Since we had a long summer break this year, and now we are back to organize. We organized 4 sessions for the last quarter in 2018, and we'll start with an external lecture. You may know Lone Farm. She has already presented 2 sessions here in the PML School, and she will give today's session, which is a really exciting story and a nice application combining population PK analysis and bioequivalence and simulation. So this is really a nice story, and which Lone will be presenting. But so that you know, I mean, there will be 3 following sessions on a bi-weekly basis, where I'm going to present PD models with non-continuous data. So please stay tuned. And you know that due to the new legislation, we are not allowed to send out email blasts to invite people, so you would need to rely on information either on our website, Certara homepage, or through our LinkedIn group, where I'm typically posting the events, and when you have a chance, just register for those events. But today we are we are listening to Lone Farm's presentation, and as usual, you can use the Q&A panel on the right-hand side of the WebEx console if you have comments and questions. So we have a Q&A session at the end where we can answer those questions. So, but for now, I guess it's time for Lone. If you can share your content to the public, and then you can go ahead with your presentation. Thanks, Lone.
Thank you, Bernd.
Yeah. Right.
Showing my screen. Are you seeing my screen now? Okay, good. Good morning, everyone. Thank you, Bernd. Not sure it's good or bad to present the opening session for this fall, but here we go. This session is not intended to only focus on how to perform population PK modeling using Phoenix modeling language, but also to emphasize where population PK modeling can be used in the process of formulation optimization or at any point of drug development. This example presents to you the results from an integrated scientific team that strategically selected a creative, unconventional approach to develop a successful formulation. After this session, the take-home message today is that we can be very creative in any phase of drug development, including formulation optimization, and Phoenix has a variety of tools available to accomplish your creativity. Personally, I am a huge fan of Phoenix and have been using the tool daily as it fosters my accomplishment. With that, first in our agenda today, we will go through an example, including available data, steps were performed to find the successful formulation, starting with population PK modeling and steps for simulations of virtual bioequivalent trials. We will have demonstration of these steps and question and answer at the end. The assumption for the audience for the talk today is that not only we are familiar with Phoenix and LME, but we also have a good background and familiar with non-compartmental analysis and bioequivalence study design and assessment, the tool in Phoenix as well. In this example, there are 2 types of I think that I jumped to, okay. In this example, there are 2 types of pellets. that are intended to produce extended release and immediate release profiles. We call them formulation 1 and formulation 2. The reference product is capsule containing modified release bits. The data available obtained from 2 studies in human volunteers. The study 1 is comparative bioavailability study, single dose, 2-way crossover, formulation 1, which is extended release bits type versus reference in 12 subjects. Study 2 was a comparative bioavailability study, single dose, 4-way crossover, formulation 2 versus reference versus 2 prototype of the formulation in 34 subjects. From these studies, the estimated intra-subject variability of this drug is less than 30%. And power calculation for a bio trial that has sufficient power of the study will require around 34 subjects. So the mission of this example is to find the right ratio of Form 1 and Form 2, the right ratio of the IR pellets versus and the ER pellets for the final formulation, which is a capsule that contains the IR and ER components that is able to then demonstrate the bioequivalence to the reference product on a single dose PK parameter, Cmax, AUC last, and AUC infinity. This is the results summarized, focusing on the test product pellets, IR and ER, and reference product. So on the plot, you can see that the ER, the extended release pellets, exhibited a delay in Tmax and lower in both Cmax and total exposure is around like 23% less compared to the reference product. However, the IR formulation show a greater and faster in vivo absorption. Cmax is almost 2 times higher than the reference product. The total exposure improved around, if you're looking at this number, around 25% compared to the reference product. And overall, we have 45 individual plasma concentration profiles, 11 with extended release for bits pellets, and 34 with the IR pellets. To find the right ratio of IR and ER for the test product to be bioequivalent to the reference product, the first step for simulation of virtual B trial is example is to establish a population PK model. Followed by step 2 is to simulate based on the established pop PK model, we simulate individual plasma concentration of the test product, that is from a test, a capsule containing the IR and ER, you change the different ratio of those 2 pellets type, and then we assign sequence and period for the test and reference in the virtual trial to the test and reference treatment, followed by the NCI analysis and the bioequivalent assessment. Now, the right ratio for the IR and ER in the capsule of the test product, virtual capsule, should demonstrate the bioequivalence to the reference product for Cmax, AUC last, and AUC infinity. So let's start with step 1. There are multiple steps as usual when we perform population PK modeling and simulation. This is a summary of the final model. The data is get fit to a 2-compartment model, log additive as error model. So we fit simultaneously a population PK model to 45 individual plasma concentration profile of the IR and ER, Form 1 and Form 2. And formulation is set as categorical covariate or factor on the Ka. So this is the graphical model for this example. You can see that formulation 1 has the rate of absorption Ka, constant Ka, and then Ka1, and Ka2 is formulation 2. Now, we can do the model fitting totally with this example by using graphical modeling in Phoenix, in which we can select treatment contain ER. You look at this right here, my mouse moving around the treatment right here under the covariate type. You select treatment as covariate, categorical, and you can select the IR and ER in this tab, so that this tab in the graphical model in Phoenix will only allow you to do that. You can also identify the secondary parameter using this tab. So in this scenario, we code the IR treatment of Harris as 0 and ER is 1. So what it means is that the absorption rate constant Ka in the result Theta table will be the absorption rate for the IR formulation. The secondary will calculate the absorption rate constant from the formulation coded 1, which is ER formulation in this example. So because this is for the purpose of the PML, I will use the PML code for this example. So as we are now familiar with the code, the code is to describe the two-compartmental model all around. Those input via AA column right here. Other parts of the code support the two-compartment model with log additive error model. It's right here, yeah. So I think there's one a little different from the usual code is that the covariate effect, COFKA, right here, is coded to identify that formulation is covariate effect on the rate of absorption of the model. So because like in the previous slide, we already coded the IR as 0. So the secondary code right here is to calculate the rate of absorption constant for the ER coded as 1. So by the time we finish our run, we have the result of the Ka for both IR and ER. I'm presenting several diagnostic plots to demonstrate the goodness of fit of the model to the data. The individual weighted residual versus individual prediction right here show that there's no significant thinning of the red lines here. And the individual prediction and observed data where all the points fall close to the line of unity y equal x here. The QQ plots of the individual weighted residuals show that almost all values fall roughly along a straight line of unity y equal x, and no significant deviation from normality. The predicted concentration values versus observed value plot here for subject 1 as representative, showing that the established model captures very well the data. And we are looking at the population PK modeling results. The CV of all estimate value of PK parameters are less than 14%. Very good and very good confidence with the model. KI here is the absorption rate constant for the formulation IR, and the secondary result shown here is the absorption rate for the ER, and you can see that the IR absorption rate is roughly around 7 times higher than, faster than the absorption rate constant value for the ER. We also run bootstrap simulation to validate the model that we established. The model predicted values here. I put those side by side with the bootstrap on the right hand. The predicted value from the model should fall within the 95% confidence interval of the bootstrap runs, and all of them fall within the 95% confidence interval of the bootstrap simulation. The secondary value K from 1 was calculated from all bootstrap replicates, and it is almost identical to the estimated value from the model. So the step 2, so we established a population PK model. The step 2 is we want to use the model to simulate the plasma concentration of the test product. We do it by accept all the fixed and random effect values from the model, and we will demonstrate this in the demo section. But in the code, you just put it in right here for the code like we showed before. So all of the values that we obtained from the run, we just put in the coding, the PML coding right here. So the step 3 is what? The step 3 is now that we have plasma concentration of the test product. And of course, when we simulate it, make sure we simulate the set of data from the same size of sample size of the reference product, and we assign sequence and period to the subject. We call it post-hoc assignment. You can utilize the sequence period of the existing study that you performed, which I use in my example. And then you run, first you run non-compartmental analysis and followed by bioequivalence assessment. So the results from our research show that if the ratio of ER and IR is 90 to 10, we are able to achieve bioequivalence to the reference product for all Cmax and AUC infinity. Now I would like to walk us through the demo session. So the first step is to so I want to show the graphical model that you can definitely run this type example using the graphical mode only. And you can choose clearance as the domain for the PK feed, extravascular, and then full compartmental, log additive as error model right here. As a parameter. From here, you can choose the covariate type, select treatment, and then you allow arbitrary category names, convert it to 0 to 1, and then before we do that, you should note that when you select covariate type treatment right here, there is a treatment column up here in the main domain. So you need to remember to map it right here. And the other part you map as usual. And then, of course, you need to take care of initial estimates to make it easier for the model to run well. Well, it takes time here. Oh, it's weird. It doesn't matter. So let's look at the textual model. Now, but since I opened the initial estimate, it takes time to go back. So this is how the model looks like. And if you start with the, as usual, if you start with the graphical model, it will be very easy for you to do some modification to the text. So what I did here is I add the secondary calculation for the rate of absorption of the formulation 1, which is the ER formulation. So what else do I need to do? But that's it. So we just hit run, and here's first we go to the theta model, the theta tables to see all the results that I just showed you in the slide deck before, and then go through all of the diagnostic plots. Let's see, where is it? Let's this is the one that we showed before, and see individual fit. Let's see, where is that? Let's go here. So this is my favorite part. After I finish the run, I want to go through each individual to see how well it captures the individual observed data. So let's see, after we are done with the model, we want to see the observed and predicted. We want to copy the model down and accept. This is a step that I mentioned in the slide before. We copy the model and go to the parameter tab right here, check all the estimate right here and accept all. So when you accept that, it will appear in your code that you accepted all the fixed and error effects and parameter as well. And then you want to change in the run option, you want to change it to the simulation. And in the tables, I think Bern showed us so many times before, we want to add a simulation table 1. And the data time points can be flexible, selected by you, whatever you want. Like this one is from time 0 to 6 hours. I want the plasma formulation simulated every 1 hour and then follow afterwards the way I wanted it. Then when it's done, you just send the table 1 by the simulation tab 1 right here, send it to the plotting, and you can see the observed versus predicted for each individual. You see how well the data capture your data. So that's the step that you want to make sure that the model is doing all right. So when the model is established, the next step is to do the simulation for the test product. Now, in order to do it, if we remember the slide, the IR palace demonstrated or exhibited a very fast and greater rate and extent of absorption, and we can do some educated guess that we will need a little bit of the IR to combine with the ER so that we can balance it out to have a formulation that can demonstrate bioequivalence to the reference product. So when you see that, you can change you keep the total dose, you change the ratio of the dosing for the formulation 1 and formulation 2 from, let's say, 90% to 10. You can start with 85, 35. So 35 is the IR. So you just keep doing it, but just after a few trials, you already get the ratio that allow bioequivalence demonstrated between the test and the reference product. Now, how to do it? It is all about the dosing column right here. So I tentatively show you the time column at 0 hour right here, because it's where you put the dose in, and right now it is by the time this is the observed data. We dosed both IR and ER 400,000 unit dose, but then for the purpose of simulation, you want to, let's say you use 400, and if it's 10, it should be so 360 versus 40,000, meaning 40,000 unit dose for the IR, and then 360,000 unit dose for the IR. So you just put it in here and you do the simulation. After you obtain the plasma concentration of the test product virtual, you want to plug it back in to the bioequivalent trial. I usually use the existing bioequivalent trial because it already have the sequence and period. So what I'm doing right now is exactly the sequence period assignment for the study 2, where I have 4-way crossover study, and I just keep them here. So I just plug it in. And after you plug it in, you first run the non-compartmental analysis, and you send the final parameters pivoted to the bioequivalence assessment, and use reference formulation as a reference product, reference treatment as reference formulation in this assessment. And just extracted out the B table. When you extracted it out, you only care about the test and the reference result assessment of the result right here. So the ratio shifted to closer to 100%, and the 90% confidence interval for the geometric mean ratio of both of all Cmax, AUC, last, and infinity fall within the bioequivalent limits of 81-25%. So that's how we obtain the final model, the final ratio of the test formulation 1 and 2 that can demonstrate bioequivalence to the reference product. So so that is how all the steps we needed to go through to accomplish the mission. I would like to go back to the slide right here. So to summarize this example, we utilize the population PK modeling to establish the best PK model with the very high confidence of the model that the simulated plasma concentration for the test product should be very reliable. And then we help the formulation team to identify the ratio. The next step is to enroll 34 subjects for a pivotal bioequivalence, and conducted to demonstrate bioequivalence for the submission trial. So today example is just to demonstrate it to you that in order for us to have a biopharmaceutics research success, we need an integrated team, including a formulator, clinical expert, PK scientist, and importantly, Phoenix, a powerful PK/PD software. This approach allows you to be very flexible on the drug target product profile, where various factors need to take into account, including clinical implications such as the impact of safety and efficacy of a drug product when you intend to make any modification to the in vivo drug release profile. And again, Phoenix software is very flexible and allow you to do that upfront, proactive, when there is a biopharmaceutics project fall into your department, you really need to have an integrated team to figure it out. Now, IVIVC approach is the best, no doubt about it, but there are so many ways during the drug development process you can be a little more creative and figure out the best way, most efficient way to accomplish the task, the mission of the biopharmaceutical research. By that, I am ready for questions. Any questions? Burns?
Yeah, hi, Lone. Thanks, first of all, for the nice lecture. It was really exciting stuff. Cheryl, I don't see any questions right now, but maybe one question. One question, Lone. I guess this is a very interesting approach and application that should actually happen, more often when you are dealing with formulation design. Do you see that in the practice as well, or is this something only a few people or a few labs are doing?
So you can see that in this practice, we do make modification to the drug release profile, and depend on the purpose of what we want to do. If you really want to just have the bioequivalent trial of a product via 505(b)(2) pathway, so when you demonstrate bioequivalence on the critical components, most of the time it's Cmax and AUC, and sometimes it is early partial AUC. This simulation can predict the likelihood of passing bioequivalence on any PK bioequivalence criteria. So that it depends. So in some other cases, you may only want to take care of to be bioequivalent on a certain areas of curve to ensure efficacy, but because you don't intend to demonstrate bioequivalence on every single PK parameters or PK bioequivalence criteria, you may not obtain either safety or efficacy data from the reference product. So then it depends, but it can be widely used in a 505(b)(2) product pathway.
All right, and maybe one other more practical question. You have limited the simulation to these ratios of 90 to 10, 80 to 20. I mean, is there any desire to nail it down to the first decimal place or something like a ratio of whatever 89.5 to a 90.5 or something, or is that not practical?
Yes, but I noticed something is that if the bioequivalence is too sensitive to the ratio of the IR and ER, we may run into the issue of some errors from simulated data versus observed data later on in the trial. So it is the best to have a wiggle window, if you will. So I did several tests. I said, well, if it's so sensitive to the dose, if you change a minimal in ratio and it's just outside the bioequivalence window, that's not a good sign, right? But we did, yeah.
All right. All right. No, thanks very much, Lone. Do we have any questions from the audience right now?
I do not see any questions.
Okay. All right. Then let's close this session. Lone, can you do me a favor? Yes. Thanks first of all, Lone, for this once again for this nice lecture. Coming up next time, I mean in 2 weeks, there's the next session of our PML School series, and we are coming back to the translation of popular non-MEM models into Phoenix and LME PML code. And this is our first example where we use non-continuous data, PD data, categorical response analysis. So this is happening 2 weeks' time, October 25th at 10 a.m. Eastern. Please register for this webinar if you haven't done so already. And I'm looking forward to see you there. Thank you all for joining, your interest, and we'll see you then in 2 weeks' time. Thank you. Bye.